So good morning, everybody. I make it 10:15. So thank you so much for coming to Symposium 30, where we're focusing on CFTR. And so we've got 4 presentations to explore mechanistic insight into CFTR function and modulation. So Professor Baer and I are delighted to sort of welcome you to the presentation. So just sort of... he said, if we can get that, no, can we get the next slide? Oh gosh, this is embarrassing. Right. Oh, there we are. Next slide. So Professor Baer kindly asked her colleague, Professor Ratchan at the Hospital for Sick Children in Toronto for a quote as to why we need new modulators. And so you can see here, so while most people with CF now derive significant benefit from current CFTR modulator therapy, not everybody shows the extent of clinical response, and some individuals, unfortunately, have significant side effects. So we also do not know whether the extent of improvement in CFTR function seen with current modulators, while impressive, is sufficient to halt disease progression. So therefore, it's still an unmet need for new CFTR modulators, and these modulators could benefit those individuals which I've previously mentioned, or and also that enhance CFTR function beyond that seen with currently available drugs. So without further ado, I'd like to sort of just list the presentations. And so I'm not going to sort of read out the titles, but I just want to draw your attention to the unfortunate fact that Professor Vergani has a family medical emergency, and so she's had to go back to Italy at very short notice, and so so she's not available to give her presentation, and so instead I'll talk about some studies that my colleagues have done, or rather my colleagues have undertaken using elexacaftor, tezacaftor, ivacaftor. So without further ado, I'd like to call upon Han Yi. So her presentation, Mechanism of Action of X316761, a highly efficacious potentiator studied using structural biology and electrophysiology. So there we escape. There, start. Okay. There you are. Right. So thank you.
Thank you. I'm Hani. I'm very grateful to be here to show you this study of a highly efficacious new potentiator X316761, study using structural biology and electrophysiology. I have a disclosure. Part of this work was supported by AbbVie. So as you all know, that CFTR potentiators are compounds that can increase the activities of the channels that are already expressed on the cell membrane. And the Vertex compound ivacaftor and the Galapagos compound GLPG1837 are perhaps the most widely studied and used potentiators in our field. And although GLPG1837 is not used as a treatment for patient, it is very useful in basic research because of its high efficacy. Most of the time, it's higher than ivacaftor, and also because it's very easy to wash out in electrophysiology experiment. And I will mention this potentiator multiple times, so I'll just refer to it as GLPG. And now I am going to show you this new potentiator X316761. It is chemically, structurally unrelated to the existing potentiators, and we believe that it has the potential to outperform the existing potentiators. And I will just, for simplicity, refer to it as 761. So first, let's see how it performs on G551D CFTR. Here we are looking at a excised in-cell patch, a recording of an excised in-cell patch containing G551D channels. And because it's G551D, its open probability is very small. We have very tiny currents here in ATP. then we add only 1 nanomolar of 761, we already see a big rise in currents. Then the current rises dose dependently, and that gives us a dose-response relationship of 761 with a EC50 around 100 nanomolar. For comparison, GLPG on G551D has a EC50 around 1 to 2 micromolar. So 761 is 10 times more potent than GLPG. And more importantly, the maximum potentiation by 761 on G551D, or the efficacy of 761, is 60-fold. In the same experimental setting, we see GLPG has a efficacy about 35-fold, and ivacaftor is 10-fold. So 761 is really the most efficacious potentiator we've seen so far on electrophysiology experiment. Similarly, on F508del, we see that GLPG gives us current about this high, and then 761 is more efficacious than GLPG. And again, most cases, GLPG is more efficacious than ivacaftor. And perhaps most encouragingly, in the gating mutant we've tested here, 761 has a maximum fold increase or efficacy that is either superior or comparable to GLPG. So we have this very powerful potentiator now, and we want to know what's the molecular mechanism behind it. We carried out a series of experiments, and to save you some time, the bottom line is we find that 761 shares the same mechanism as GLPG and ivacaftor. 761 is effective on not only the gating mutants I've shown you, but also hydrolysis deficient mutants, delta R CFTR, delta MBD2 CFTR, and it potentiates CFTR gating, both the ATP-dependent and ATP-independent gating, primarily by prolonging the open time of the channel. And most informatively, or most crucially, 761 does not work on mouse wild-type CFTR. That is a characteristic shared by GLPG and ivacaftor. GLPG and ivacaftor do not work on wild-type mouse CFTR, but a single mutation in their binding site can restore their action on mouse CFTR. And we've seen that with 761 as well. So all these similarities shared by 761 and the two other potentiators lead us to the hypothesis that 761 and GLPG might compete for the same binding site on CFTR. To test that hypothesis, we carried out this classic competitive experiment, which is based on the idea that when two drugs bind to the same site, the occupancy of one excludes the occupancy of the other. So here we are again looking at a electrophysiology recording. We are using delta MBD2 here, but we've seen same thing with G551D and wild-type CFTR. We first add GLPG alone or 761 alone to have the maximum effects as a reference point. Then here in the presence of GLPG, we added 761. You see the current increase slightly, but does not reach the level of 761, the full effect of 761. The simplest explanation for that is that because some of the channels are already occupied by GLPG and they are not available for 761 to potentiate. And it's because they are competing for the same site. When we wash out 761, that effect is reversible, as you can see here. By the same logic, when we have the two potentiators mixture here, GLPG and 761, then we remove GLPG. We are washing out GLPG. the current increases to the level almost about the same as 761's own potentiation. And that is because now that we don't have GLPG in the solution, all the channels can be potentiated by 761. Just let me, okay, never mind. I was hoping to make this cursor visible all the time, but. So now with this functional experiment that supports a competitive mechanism for GLPG and 761, we naturally ask, can we have structural evidence for 761 binding to the same site? Sure we do. We determined the cryo-EM structure of the phosphorylated ATP-bound E3071Q CFTR in complex with 761. The gray density is CFTR itself, and this extra pink density corresponds to the chemical structure of 761. And this model we built based on electron density map, you can see better that this extra density here is in the transmembrane domain of CFTR. When we zoom into this region, we see this binding pocket formed by TM4, 5, and 8. That is the so-called potentiator hotspot where GLPG and ivacaftor bind to, as studied by Dr. Ju Cheng's lab in the past. So on the right here, I'm showing the surrounding residues that are interacting with this purple 761 chemical here. Most of the interactions are hydrophobic. You see a lot of phenylalanine and leucine here. We also have this pi-pi stacking between the drug and F312, and also an important hydrogen bound between the drug and serine 308. These two interactions are also present in ivacaftor and GLPG. Now we have this cryo-EM structure showing the interaction between the drug and CFTR. We want to validate whether these interactions are relevant in functional studies. And also we want to know how each interaction plays a role in the efficacy and potency of 761. So we carried out dose response experiments with mutagenesis on each residue. Let's first look at the serine 308 that forms a hydrogen bond with the drug. We break that bound by introducing alanine substitution on the background of G551D. You see this S308 current need 5 micromolar of 761 to start seeing some potentiation. And the maximum fold increase was only a couple fold. Remember that on G551D single mutant, the EC50 was 100 nanomolar, and the maximum potentiation was 60-fold. That means this breaking this hydrogen bound drastically reduces the potency and efficacy of 761. That shows us the importance of this hydrogen bound in the action of 761. And next, let's look at this pi-pi stacking with the F312A mutation. We have a rightward shift of the dose response, and that is a 10 times reduction in the potency of 761. So we carried out same dose response experiment for each amino acid that we saw in the cryo-EM structure interacting with 761. And for example, the tyrosine 304, leucine 926 mutations on them all reduce the potency of 761. And I'm not showing you all the dose response just to keep my slide clean, but we are able to confirm that the interaction we saw on cryo-EM structure are indeed functionally relevant. And in addition to the potency of 761, we also want to understand how these interactions contribute to the efficacy for 761. But the story is a bit more complicated. You see that with alanine substitution, the effect is different for different amino acid. On tyrosine 304A, that does not change the efficacy for 761. But for S308 and F3128, we do see a big reduction in efficacy for 761. Then, interestingly, when we do L926A mutation, that slightly increases the efficacy for 761. The reason behind these changes are far more complicated to understand, and we can go into details afterwards. But it's more difficult to explain because unlike potency, where you break the bound, you lose the interaction, and you have a reduced affinity. With efficacy, we have to introduce this state-dependent binding idea to understand them. So nevertheless, these informations are useful if moving forward, people want to do structural-based optimization on potentiators in these binding sites. You do want to understand how the drug interacts with each amino acid, and how those interactions determine both potency and efficacy for the drug. A short summary here, I've shown you both the structural and functional evidence for 761 binding to this potentiator hotspot, and each residue contributes differently to the potency and efficacy. Then when we look more closely into this binding site and the binding post of 761, we notice this unique extension here, this extending arm that's interacting with these three residues here. This is unique to 761. If we have TM8 helical break here as a landmark and compare its binding with other potentiators, you'll see, for example, GOPG, it's sitting in the same binding pocket, and these interactions like tyrosine 304, serine 308, and phenylalanine 312, these, we call it the core interactions for this binding pocket. These interactions are shared by all the potentiators in this binding site. But GOPG is more confined inside this pocket. It does not have that extending arm, if you will. Similarly, a potentiator developed by Dr. Ju Cheng's lab using computational drug screening, targeting this binding site, has the same feature. It's only confined into this pocket without engaging extra interactions up there. Just for your reference, this potentiator has comparable efficacy with GOPG. And finally, we have ivacaftor. Its geometry is kind of interesting. It's like horizontally inserting into this binding site. But again, you'll see that it's only in this pocket. And we are fortunate to have 761 and study with it with cryo structure, because without 761, we would never think that this binding pocket for the potentiators actually has the potential to have all these extra engagement. And we know that 761 is the most efficacious potentiator so far. And perhaps by engaging all these additional amino acids, that's why it becomes the most powerful potentiator. So for future studies, especially for structural-based drug design, we should probably leverage these interactions to design more efficacious potentiators. So finally, in conclusion, I hope that I convince you 761 is effective across a broad panel of class 3 gating mutants, including 508L and G5I1D. And 761 is not only the most efficacious potentiator reported today, but also it achieves a nanomolar potency, and that is important for translational application. And 761 binds to the potentiator hotspot but it also extends beyond that region and engages more interactions with these amino acids above that TM8 helical break. And finally, 761, we hope it really holds the potential to give us next generation therapeutic candidate. With that, I would like to thank my lab members and all the collaborators in AbbVie and University of Alabama, Dr. Cappy's lab, which is supported by the foundation, help purify the CFTR proteins that we used in the cryo-EM studies. Also, we would like to thank the cryo-EM Center at New York and also at our university. And here is the funding for the study. Now I will take questions.
Excellent work. I was wondering, you must have tested this potentiator on rescued delta F also. When we rescue delta F with elexacaftor, tesacaftor, and we add chronically ivacaftor, we reduce the amount of protein, like it's destabilized and internalized. GLP-1837 doesn't do it, so I was wondering whether you knew how your potentiator acts on rescued delta F.
On cell line patch clamp experiment, the potency of 761 alone without the correctors on delta 508 del is about the same as GOPG. But I don't have the number on the top of my head, but it's about the same. We do not have patch clamp experiments with delta 508 pre-treated with correctors. 761, as I show, is binding to the same site and works through the same mechanism. As you said, that GOPG does not have that problem, but ivacaftor does. So it becomes hard to predict what would be with 761, but certainly that's a very great future study direction.
And if I could add another question, I'm just curious how this compound was identified.
I will have to defer that question to CF Foundation, which now owns the property right of this compound.
Okay, thank you. May I ask that the people asking questions identify themselves and their host institution, just so everyone knows? Please go ahead.
Sure. I know this is not easy to do, but have you attempted in any way to look at the lipid dependency of these compounds and whether there's any difference in the lipid dependency between the different analogs?
We do not specifically look into that. If it's relevant, we do compare the hydrophobicity of the drug. The most hydrophobic is ivacaftor and this one, and then GLPG. So we are thinking that the potency of these drugs also follows that order, and it's the hydrophobicity that's related to the potency.
Okay, thank you. Please go ahead.
Hi, I'm Celeste from Stanford University. I was just curious if anything has been done to know the toxicity of this potentiator or anything about the pharmacokinetics about it.
So I do not do the experiments on like toxicity and pharmaco. Is that word?
Pharmacokinetics.
But our collaborators at AbbVie and I hope Foundation in the future we will provide that information.
Thank you.
Hello, I'm Cedric Govas. I'm from Free University of Brussels. So you hypothesized that the extension of the binding site might be why you have a more potent, but I don't recall, did you mutate those or do you have analogs that are lacking this extension to confirm that hypothesis that this is where you get the extra potentiation?
Okay, so let me respond to that with two, the explanation will be twofold. First, you asked about any derivatives that lacks that part of, no, we haven't done that. But secondly, we do test those residues, mutate them one by one. Mutations on these three individually all reduce potency. So all of them contribute to binding, contribute to affinity. On efficacy, it's more complicated. I've shown you that L926A increases efficacy. Threonine 925A does not have effect on efficacy at all. F315A mutation does decrease efficacy. So for efficacy, the change is different and all in different direction. Do I still have time? Okay. It's hard to explain why structurally, because for efficacy, that potentiation comes from the interaction of the drug and CFTR in both open state and closed state. You need to understand how the drug binds to the open state and closed state and what's the difference between them to explain, because potentiation is state dependent. And so far, we do not see a difference between drug binding to the open channel conformation, drug binding to the closed channel conformation. And that's because first, we don't have a really open channel conformation in cryo-EM structure. But yeah, experimentally, we've verified that these 3 residues contribute to potency. All of them contribute to potency, but they affect efficacy differently. I don't know if that kind of answers your question.
We still have a couple of minutes. If I could ask a question, could you comment on the effect of the potentiator that you're describing, 761, on different variants, disease-causing variants? I think you mentioned some in your poster. It would be good to maybe summarize how it's working on some of these others.
On 508del, on G5RYD, and N1303K, 761 is better than GLPG. Then we've also tested R117H, even though it's known as the class 4 mutation, it also has gating defect. There's no difference between 761 and GLPG. Then on L927P and G1349D, we don't see a difference. Why we don't see a difference or why we do see a difference, I don't have like a structural explanation for that.
Okay. Could I follow that up? So have you tested variants that are insensitive to ivacaftor? So I'm thinking about variants like R347P, where when you have that variant at the cell membrane, you don't see potentiation with ivacaftor.
I have not tested that, and I can see now I'm going back to test it. Yeah. But we would expect that because they bind to the same site, probably. If it's insensitive to ivacaftor, it likely be insensitive to 761. But I'll have to test it.
Be worth a test.
Yes, it's worth the test.
Okay, well, thank you.
Over to you then.
Thank you. Well, first off, thanks to Christine and to David for giving me a nice excuse to enjoy for the first time the rain here in Seattle. We have excellent rain in Brussels, but this is even much better. So the premise I want to I just want to remind everybody, we all know that CFTR is an ABC protein, right? It's not a transporter like all of the other ABC proteins, but it has the same architecture. So you have these transmembrane helices that are connected to the NBDs. And the common model is that in order to alternate between the closed state and the active state, these two NBDs, they have to come together. And the ATP will be the molecular glue. And once ATP is being hydrolyzed, and then the molecular glue is released, and then the two NBDs can separate. And so the association of these two NBDs seems to be strictly linked to the conformational change at the level of the TM helices to form the ion conduit. So this is really the common model that we all accept, I think. And I would like to slightly challenge this as being the unique way of activation of CFTR. With this in mind, I don't need to remind you the key questions that remain. I think David mentioned the first, the need to get better modulation or improve current modulation to adapt to other non-responding patients. But as a I will briefly touch on that, or I will touch on that, but also as an academic, I want to extend our understanding of the activation mechanism, as I just sort of alluded to. So we all know that, the prototypical class 2 mutations, 508, DEL, and others, they are characterized by a thermal destabilization of the protein. And we know from many studies and many years of studies that thermal stabilization of NBD1 should be highly advantageous in a therapeutic approach. And obviously that's what Siona is trying to do right now. But we had decided in my lab to develop specific CFTR stabilizers to try to probe that hypothesis further. And the way we had done it was to develop nanobodies. So in case you're not familiar, there are mini antibodies that you can get from llamas or camels because they produce these single chain antibodies, and you can isolate these mini antibodies called nanobodies. And we have a collection of about 50 or 60 of them in my lab that all recognize NBD1 of human CFTR. And we had shown that they can bind with high affinity, the isolated domain and the protein, the full-length protein. Then they can thermally stabilize the isolated domain, but also the full-length protein. And we had done co-crystallization of the NBD1 and those nanobodies. So we have a pretty deep understanding of the epitope, pretty much at the atomic level. So with these tools in hand, we wanted to see if we could use them as chaperones to improve the response of our beloved modulators, in the case specifically here of F508, but now we're trying to extend that to others. So what we were lacking was a relevant delivery method. And after COVID, of course, everybody went into mRNA LNPs. We did as well. So now we can get our nanobodies inside cells by just getting mRNA into LNPs, getting those LNPs onto cells. And what you can see on this slide is that if you have here, you have two examples of cell lines, HEC and TFBEs, where you have a moderate response to ETI. But in these cells, if you add expression of the nanobody, you have a huge increase in band C, right? And we have this here in this case with HEC. You have this case here with CFBE. We can also use an extracellular engineered epitope to show this by fact that you have quite a significant increase in the expression. I was expecting that, but I was not expecting an effect on function. Still, we tried. And in collaboration with Marcus Moll and Tihomir here in the audience, I think, made those experiments, they were able to get these nanobodies inside monolayers of CFBs and test transepithelial current. And what you can see is that the nanobody itself has a small effect on a 508 DEL. But if you rescue it with ETI, now the combination of both of them is highly synergistic, right? So you have a huge increase in the response of these mutant cells when you combine both. So clearly we have a combination because the two modes of action are highly different. We know that the ETI and the two correctors do not thermally stabilize the protein, and we know that the nanobody do. So the combination of these orthogonal methods proves to be synergistic. We can also show this with other methods such as YFP assay. The problem is that I just told you I was expecting this to be an inhibitor. And why? It's because we had the crystal structure of the isolated NBD1 bound to the T2A nanobody, our favored stabilizer. The problem is if I superimpose this on the structure of the dephosphorylated human CFTR, the Ju-Chan structure, you see that this predicts that the T2A nanobody would just be right in the middle of the two NBDs. In that situation, it's impossible for the two NBDs to dimerize. And therefore, you would suggest that it's impossible for the channel to form. But we do see function, we do see improvement of CFTR function. So something for me didn't make sense, right? So thanks to David here in the audience, we were able to go into single channel level. And so this is a typical trace that David got in his lab. And you can see regular opening. This is a minute time scale, regular opening. The opening probability is about 0.3, 0.4 here. Now, if you add to that prep, to that patch, the T2A, you see a dramatic change in the gating behavior. Let's look into slightly more detail in the time scale. You really see these long closures that you never see in the absence of the nanobody. So that really matched the idea that the nanobody would just sit between the two NBDs and act as a inhibitor of the channel activity. But then David's group looked into F508. So this is without the nanobody. This is, you see, you have a little bit more than one channel here. So that's why the traces are a little bit more fuzzy. But you see that you can rescue with ETI and you go to 37 degrees and you have a nice channel current in this patch. But that will die off after a few minutes once you bring it back to 37 degrees. I think David will show other data later on. Now you do this in the presence of the nanobody, you don't see that decrease. You don't see that thermal destabilization. So the nanobody thermally protects the protein quite nicely. right? So two things. First, it's there and it thermally protects it. But not only it's there in binding, but it doesn't prevent the channel opening. Now, David looked in a bit more detail. And when you do, low pass filtering, you start to see next to the full deflection, you start to see these shorter deflection, which are characteristics of subconductance. So in addition to full conductance state, we start to see more and more, and you can see if you compare the F508 without the T2A and the F508 with the T2A nanobody, you have more and more. You can do a histogram where you have the fully open and fully closed state, and you can start to see in the histogram more and more of these subconductance state in the presence of the nanobody. So that's probably part of the puzzle. And you can even do that in the context of the wild type. And while in the absence of nanobody, you barely see any of these low conductance state. In the context of if you incubate those with T2A LNPs, you start to see them in the wild type as well. So there, it was really hard for me to understand. And so turned to John Hunt here also in the audience and sent him some nanobody. And he was able to get the high resolution structure of the complex between wild type. It's important to keep in mind it's a wild type human CFTR and T2A. And he found mostly two conformations. I'm only going to talk about these two conformations. So the first one we can see from the envelope looked exactly as I was expecting, right? So you can already see the V-shaped inactive state of the protein. So we could build the model very easily, and you can clearly see you have the V-shape, inverted V-shape, NBD1, NBD2, highly separated. The channel must be closed, and you can clearly see the position of the T2A nanobody, and it's exactly where we predicted from the crystal structure. So there's no surprise whatsoever there. And actually, if I superimpose this structure with the original chain structure of the non-phosphorylated, they are basically identical. There's a little bit of motion in the NBDs here, which is required to make room for the nanobody. But basically, the channel part, the helices, are absolutely identical. So that's a closed state, and that matches what we expected, with the long closures of wild type. But there's another conformation that Johns was able to identify, which covers about 1/3 of the particles where clearly he could see a T2A. And that was quite a surprise. You can really see from these images right there that something is happening at the level of NBD1, which is very different from what we've seen. And you can see from the density here that it's completely affected, dramatically changed from what we originally expected. But importantly, even though the density is not as good as the one we had in V-shape, you can clearly identify the different elements, and we can build, in an unambiguous manner, a model of CFTR. And you can see here that this results in an NBD1, which is relocated towards the membrane. So it has undocked from the ICLs, and now it's located towards the membrane, right? This was not expected at all. Well, that's some ideas that undocking, actually, now 219 papers, we had predicted that we would have some undocking. But this was the confirmation. Now, what does that mean from a functional standpoint? So first, if we look at the left-hand side, you see that the TMD2 NBD2 part is identical in both conformations, the conformation in the V-shape and the conformation where the NBD1 is undocked. They're pretty much the same when I superimpose them for the TMD2 NBD2 part. But if we look at NBD1, we see here comparing the light blue and the dark blue, we can see this huge motion, right? So there are two rotations, one like this and one in the y-axis, that bring the nanobody really to the where you would expect the membrane to be. So that's a huge change, but it's accompanied by a motion of the TM region. And if you really look, if you compare the gray one, which is the V-shaped one, with these green helices which come from this alternative state, you see that the helices come together. They really change. They move relative to each other. And what that does is that it actually creates the ion conduit. If I superimpose now the green helices coming from this alternate structure to the phosphorylated structures, which is as close as we have, I think, from an active channel, they are pretty much the same. You see that, of course, here the NBD is not there. It has moved away. But the rest of it here is pretty much the same. So this indicates that in this state, the chloride channel is being, and we would hypothesize that that's an active state that could correspond to the T2A bound active activity that we've seen. But if you look a little bit more in detail, actually the density is not perfect everywhere. No, it's not perfect nowhere, but it's even, I would say, poor in some regions. So the density around ICL4 and ICL1, basically what you would expect, the NBD1 to come and to lock, you see that the density is poor, the density is lost. What our interpretation of that is that these regions are moving. So if these regions are moving, you would lose the density in the crowd. But also, you would expect that several conformations are possible for this region. So if that's the region which is involved in forming the channel, you could imagine that the conformational diversity that you observe here could be related to those lower conductance states that we have observed, that David basically has observed in this activity. Now, you can say that, of course, this is only due to the T2A nanobody. It's not biologically relevant. It's an artifact. But we didn't think that. And so what John did was to look back into grids where he had wild-type CFTR without the nanobody, using the information we have, which is what kind of conformation we're looking at. And on the top here are example of 2D classes where we see the NBD1 here being undocked next to the nanobody in grids where the protein was incubated with T2A. Now, if we look at grids where the wild-type CFTR is not incubated with T2A, you do find some particles. They are rare, but you can do 2D classes which are highly convincing. You do find these NBD1 moved away, undocked, and going to the side. So you don't need the nanobody to actually have that state. It's actually relevant as far as the cryo-EM grade can be biologically relevant. You can actually find it in those grids. What the nanobody does is not inducing this state. It's just shifting the equilibrium. So we have more particles in that state, in those grids that contain the nanobody. It enabled us to build the model and the structure. And therefore, this is how we can now go back to the other grids and find it, even though it's a rare occurrence. So what I would like to sort of take home message here from a more structural and fundamental side is that we entertain the idea that there's more than meet the eye when it comes to active state of CFTR. The nanobody probably has enabled us to uncover new states, and I would challenge the idea that this is the only active state, but rather we have to consider a diversity of states of CFTR, including some active states. And with that, I want to thank the people that have contributed to the work in my lab, in Marcus, John, Dave's lab, and everybody around that has been very helpful in getting this quite amazing set of data together, and also all of the funding buddies. I've talked fast as usual. Thank you.
Joe Van Ziem from Emory and Georgia Tech. So I'm an immunologist, so I'm coming from the angle of the nanobody itself. The complementary determining region is binding to NBD1, apparently. Mm-hmm. Do you think that there's some binding to lipid determinant or some other component of the CFTR kind of signaling platform on the other side that might actually stabilize that sideways conformation?
In the cells, I don't know, but the cryo-EM experiment is done with few lipids. It's done in detergents. There are some lipids that can stick around, but you don't see it. So you do see lipids bound to the protein. You do see cholesterol, but we don't have the platform. So we could ideally want to do experiments in nanodiscs that could change. I would not be surprised if it would change the equilibrium.
But binding to neighboring CFTR.
It could be. Would have to design an experiment to test it. In this case, you don't need a neighboring CFTR to have that effect, but it could help that.
Because you saw an increase in the substates when there were more than one channel. Is that correct?
I wouldn't say that that's the case. I don't think we can say that.
Okay, thank you.
Martina Gensch, University of North Carolina, Chapel Hill.
Hello, Martin.
Fantastic work, Cedric. It's really amazing collaboration with awesome people and how this has progressed. Structural and functional and pretty impressive. I was wondering whether your active state with the T2A bound to CFTR is also linking it back to the first presentation, sensitive to potentiations that you can further activate it.
So, to my knowledge, did we compare with and without IVA capture? All the experiment we've done, we used IVA capture. Yeah, you did. We did. So I think we always used IVA capture. So the question. So it is.
So what about without IVA capture?
I'm not sure we've compared. We could do that.
Would be interesting.
So so there would have been a control period without. Sorry, Martina.
Actually,
So in some of the experiments, there would have been a control period in the presence of the nanobody, and then ivacaftor was added afterwards.
So here you have the data. You actually this is without. Sorry, I apologize. Thank you, David. So here this is before ivacaftor, and then you add ivacaftor and you increase the open probability. So it is sensitive.
Thank you.
Yeah, great.
Hey, T.C. Huang, University of Missouri. In your cryo-EM work, John's work, and are the proteins phosphorylated?
Yes.
Is the structure solved in the presence of ATP?
Yes.
In those two conformation John resolved, where do you see the alpha helix of our domain?
No, we don't. Well, there are bits that we can attribute. Maybe it's a whole debate between John and I where we don't always agree. That he's the guru, so I cannot say anything. But no. So there are density that we cannot explain. Can we definitely attribute those to R? John can probably comment on that.
Oh, okay. All right, good.
So just so that everybody got that. So, John, please correct me. So you're saying you could see a short segment?
Yes.
Okay. 8:25 to 8:35. Okay. Thank you.
Hi, I'm Celeste from Stanford University. I have many questions about this. And I was wondering, I guess, first of all, have you looked at the rate of endocytosis of F508 delta nanobody corrected to see if it's stabilized at the plasma membrane and increasing the half-life?
We have one experiment, sort of preliminary, that suggests it doesn't. So I was super disappointed.
Okay.
So we did one experiment looking at stability, and it didn't seem to improve from that one experiment.
Okay. And do you think that when you're looking at these currents, are you thinking that the CFTR is bound to the nanobody, or if it's just the nanobodies coming in, helping it fold, and then helping it traffic, and then what you're actually reading are like properly folded CFTR, but not antibody bound? Is it like a mixed population?
So this you could interpret this as this being bound and this being unbound, right?
Mm-hmm.
But those nanobodies are low nanomolar, and we know from biochemistry that they're hard to basically wash off. In this experiment, this is an experiment that I did not want David to do, but he did anyway.
No.
In that experiment, the cells were treated with LNP. So it means that the nanobody was provided as mRNA and synthesized by the cell. Then those cells were used to do patch clamp. And then those patches were not seeing any more nanobody. So it's basically diluted in what, half a milliliter of solutions. We don't provide any more nanobody to the protein. So either it's always bound to it, or it has to stick around for some reason. My supposition is that it's sticking to the protein. I don't think it's going from one to the other.
Do you think it's worth doing cryo-EM with the F508 delta to see if it has the same?
Yeah, it would be great. Yeah, I mean, obviously. So I think we have more of these states because probably, at least in the case of F508, we have to look for other class 2. But at least for F508, probably the interface is weakened. And so you have more of these undocking events. And so that's why you probably have, slightly, I would say, a good efficacy of the nanobody. I also think one thing I didn't tell you is that the experiment where we see these substates, the subconductant state for wild type, they are not exactly the same as before. It's not the protein is not added acutely. That protein has been provided as mRNA, and so CFTR has always seen that protein. So I think the undocking, my interpretation is that in the case of wild type, the undocking event is a natural equilibrium, undock and undock. And actually, there's old cross-linking data that suggests that that's something that happens all the time. And this undocking happens in other ABC. It's actually known, but people just don't publish structures where you don't see it because they think it's trash. So these undocking are probably rarer for wild type, and that's why you need to have constant presence of the nanobody. Once it's there, it's probably going to stick to it and prevent re-dimerization.
Thank you.
Thank you.
I have a question. It may be unfair, but I'm curious if you've tried to model conduction through the pore Have you tried to model water or chloride going through the pore? And would bicarbonate go through the subconductant state?
Problem is that the resolution there is just too bad to actually. I mean, we cannot even position properly the side chains. So if I play around with the side chain, it will still fit, and I can make it go through. I've tried to really model the conduit. So I've tried. Yes, it looks like the channel is being formed. I have put a lot of condition. It looks like a channel formed. Does it really? Do we have enough space in those structures? I think the resolution is not high enough to answer that question. I mean, honestly.
The second part was related to bicarbonate conduction. So would the subconductant state mediate bicarbonate conduction?
David?
Not tested. Not tested. Yeah, it's a fascinating question. Yeah.
I'm from Emory and Georgia Tech. Sorry, I had another question too, sort of a real world question. Nanobodies are highly immunogenic, right? I mean, so if you give them in humans, you will have anti-nanobody antibodies. So do you see this mostly as kind of a tool to understand some substates of CFTR, or do you envision potential translational use?
So nanobodies can be humanized to decrease that quite dramatically, and they work, right? I mean, they're being used as drugs. Yeah. Obviously, if Siona's small molecule works, this is just a proof of principle, right? Otherwise, you can go into science fiction and trying to get it. We're going to try it just as a proof of principle to take it as far as we can into animal models and patient cells and see how far we can go with the principle. You never know.
Okay. All right. Thank you. Thank you. Okay, it's my pleasure to introduce the third speaker, Jonathan Ditlev. So Jonathan Ditlev is a scientist at Sick Kids Hospital, and he's going to be speaking about coupled protein and lipid phase separation and the modulation of CFTR clustering on membranes. So a new area that we haven't seen too much about recently. Okay.
All right. So I just wanted to start off by thanking David and Christine for giving me the opportunity to share some of our work into this new field for us. And just to let you all know, I'm not so focused on the single molecule level of structure, but rather a mesoscale structure involving probably 10 to 100 CFTR molecules organized on a membrane. And before I begin, I just want to say that I currently advise two startup companies, DuPont Therapeutics and Neurophase, neither of which has anything to do with CFTR. Okay, so cellular organization. Cells are packed, as we all know, with biomolecules. In this illustration, we can see that everything is pretty well distributed. We have nucleic acids in the nucleus, membranes, etc. But this image doesn't actually capture what's happening inside of a cell if we think about the concentrations of proteins alone. And so I'd like to give you a different example. So if we think of proteins as humans and the volume of the cell is the Great Salt Lake, all of the humans on the Earth would have to fit into the Great Salt Lake for it to be the equivalent of what's happening in cells with proteins. And so in this dense, assumedly well-mixed environment, we have to ask ourselves, how do any proteins come together to do anything? Well, one way that's well known is that membrane-bound compartments, such as mitochondria, nucleus, etc., are able to organize proteins into different compartments so that they'll function in specific goals that the cell has, such as energy production or gene transcription, etc. Another more recent development in cell biology is the non-membrane-bound compartments that we call biomolecular condensates. And these structures are also localized throughout the cell. There's been numerous found in the nucleus. There's been numerous found in the cytoplasm. And I really want to focus today on membrane clusters because these non-membrane bound compartments don't have to necessarily be separated from membranes. Instead, they're able to form on membranes. And importantly, many diseases, and more emerging every week it seems, are related to faulty phase separation within the cell. This includes ALS, frontal temporal dementia, cancer, neurological disorders, all the way from neurodevelopmental disorders to neurodegeneration disorders. diabetes, and also rare genetic diseases. And what I'd like to posit is possibly cystic fibrosis as well. And so how does this happen? Well, recently, my postdoctoral advisor actually figured out that proteins can undergo phase separation into separate liquid states that are separate from either the cytoplasm or nucleoplasm, much like you'd picture a salad dressing with balsamic vinegar and oil. And that this process underlies much of cellular organization. And so how does this process happen? Well, multivalent interactors drive the formation. And so what I mean by multivalent interactors are proteins that can interact with multiple binding partners at any given time. In this example that I'll show you are two proteins, we'll call them blue and orange, that each contain 4 domain motif interacting pairs. And 2 types of interactions can occur with this. The first is a heterodimerization, in which the 2 proteins zipper together. And this is commonly thought of when you see these types of interactions. The other is that if these domains and motifs interact with weak affinities or highly dynamic strong affinities, they can arrange themselves into a large oligomeric complex. When interactions between these domains and motifs become more favorable than interactions with the solvent, phase separation occurs, and you get two physical states within the same solution. Importantly, as I mentioned before, this can occur on membranes and in solution. Interestingly enough, it occurs on membranes about an order of magnitude easier than in solution. And so membranes offer the cell an incredible way to control phase separation of signaling complexes and even ion channel complexes. And just as examples of phase-separated structures on membranes, we have the T-cell signaling clusters, glomerular filtration barrier in kidneys, focal adhesions, Wnt signaling pathways, and also growth factor receptor signaling. And all of these occur through multivalent interactions on membranes. And so, importantly, too, these structures have been linked with many, many cellular functions. Clearly, organization of membrane-associated proteins. They can increase the probability for actin machinery activity. They can increase the probability for enzyme activity. They can control RNA translation and deadenylation, so RNA metabolism. And importantly for this talk, they can control ion channel activity, which we recently showed with Liu-Yang Wang in potassium channel organization and function. And they couple lipid and protein phase separation into functional signaling, which we recently showed in T cells. And so I'd like to think that maybe we can take the general principles that we're learning about phase-separated structures and apply it across cell types and maybe learn something about cystic fibrosis transmembrane conductance regulator biology, and maybe use that information in the future to come alongside the development of small molecules to correct CF across people that aren't able to respond to current therapies. Okay, so previous work by the Hanrahan group beautifully showed that CFTR is localized into clusters on the plasma membrane, and that their cluster formation was linked to cholesterol. And so as cholesterol in the membrane was increased, clustering went up, and as cholesterol in the membrane was depleted, clustering went down. Importantly, application of correctors also is able to restore clustering in these cells. When no drug was applied to either the F508 or S13F variants, clustering was diminished across the cell. However, application of elexacaftor and tavzacaftor were able to restore clustering, although it's difficult to see in these images. You'll have to believe the white arrows that I put on the images. It's all good. All right, so why do I think that CFTR might participate in phase separation on membranes? Well, it's because it's well known that they form supramolecular complexes on membranes with various binding partners, including calmodulin, shown here in this previous publication from Julie Foreman-Kay's group, along with Christine Baer. And these interactions depend on both phosphorylation and calcium within the cell, both of which are shown to modulate CFTR function. And interestingly, many of the binding partners bind to the aforementioned R region, this long intrinsically disordered region between the NBD domains that can apparently regulate CFTR function. Okay, so if we look more specifically at R region and C terminal interactions, we can see that there's a network of proteins that can bind to CFTR at any given time. These include SLC transporters. That interactions depend on this intrinsically disordered intervening sequence between STAS domains, as well as a C-term region that can bind to NERF, the NERF PDZ domains. The IVS has been shown to bind to CFTR region. Calmodulin can bind to both SLC and CFTR through its N and C terminal lobes following calcium binding. And NERF1 can interact through its PDZ domains with the C terminal tail of SLCs and CFTR, as well as cholesterol in the membrane through its PDZ1 domain in a non-canonical interaction motif. Okay, and so here I've got an image of the of proposed conformations of the R region at any given time. This was done by Nemo Liu, who's a student that works right outside my office for Julie Foreman-Kay. And he's shown that phosphorylation of the R region can drastically change the dynamic states of this sequence, of this peptide chain, and possibly allow for many interactions with proteins that are not available in its non-phosphorylated conformation. And so to investigate whether phase separation might be involved in CFTR biology, we use computational modeling and biochemical assays to investigate this process. And this work was done by Yimei Wan, who is a very talented master's student who's now at Yale Medical School because she chose a different line of work than what I'm interested in, fortunately. And then Dr. Rhea Hudson and Jordan Smith also participated in the study. And so the first thing that we did is we built a biochemical computational spatial model of this interacting network shown here on the left. The nice thing with building a model like this is that we're able to turn interactions on or off simply by the press of a button. So if we want to find out what calmodulin is doing in terms of forming CFTR clusters, we can turn it off. If we want to look at NERF, we can do the same thing. We can also change binding rates very easily. And so we can see if the measured binding rates, the experimentally measured binding rates, actually contribute to predicted cluster formation. And so when we run these simulations, we first turned off cholesterol in the model. So these proteins were unable to interact with cholesterol. And what we showed is that protein-protein interactions alone are predicted to form nanoscale clusters containing between 2 and 4 proteins. Larger, what we call mesoscale clusters that you could see under a microscope very easily are not formed. When we turn off protein-protein interactions but turned on cholesterol interactions, we saw the same thing. Cholesterol alone was unable in our model to induce CFTR cluster formation. But when we turned on both protein and cholesterol interactions, we saw robust clustering with some clusters forming over that contained over 100 molecules. And in these simulations, these actually used up most of the CFTR molecules. So if you can imagine the simulation space, there was a single big cluster within our simulations. But there's a diverse range of clusters that form. Okay, so this is great, but how do we experimentally test it? Computation only goes so far with modeling these types of systems. And so I previously used a supported lipid bilayer experimental system. And so what we did is we adapted CFTR to our system by generating recombinant protein that contains 2 his tags mimicking interactions with transmembrane domains. And then in between the his tags is the native R region and then the C-terminal tail that allows binding partners to interact. However, if we want to study how cholesterol can interact in this process, we need an experimental system in which cholesterol doesn't turn our membranes into solid substrates. Because as we know, bilayers are fluid, and we need to be able to mimic that biochemically. And so in the original system, anything above 5% of non-phosphocholine lipids turn the fluid into a solid. And so we need another approach to study CFTR cluster formation. And so what we've done previously is developed a double membrane system in which we can have a complex membrane on top of a simple membrane. And this allows us to vary different properties of the top membrane without affecting its fluidity. It also allows us to target CFTR to cholesterol-depleted regions versus cholesterol-enriched regions to allow us to test the effects of cholesterol on clustering. And so in this first set of experiments that I showed you, we attached the CFTR to the cholesterol-depleted regions of the membrane. And what we see as we increased protein concentration and calcium, which allows calmodulin binding, is that clusters don't form. And this supports our biochemical model that I showed you earlier, in which protein-protein interactions alone are insufficient to drive the formation of large clusters. However, when we target the CFTR to cholesterol-rich domains, what we see is that upon addition of calcium, clusters form readily. And when we quantify that, we can see that as cholesterol concentration increases, the level of clustering also increases significantly. And so these results very much support our biochemical model, in which both proteins and cholesterol are required for CFTR organization on membranes. Phase-separated structures are very dynamic. The molecules tend to rearrange. And so we wanted to test the dynamics of these small clusters on the membranes. And what we did is we looked for merging events. So taking two clusters, and do they contact each other, and do they form a new single cluster? And the answer is yes. And so our results are fully consistent with CFTR cluster formation being a phase-separated event. And the previous work across the field has shown that phosphorylation and calcium are important for function. So we asked what they do, what these two modifications to CFTR and calmodulin respectively do to clustering. And so we performed these experiments with and without calcium, and then with and without phosphorylation. And what we can see is that when CFTR is phosphorylated, we use pKa, we get robust cluster formation, but the clusters are small and spread out across the membrane. When we use non-phosphorylated CFTR, we get the formation of larger clusters when calcium is added, as I showed you in the previous images. And so all of these results are suggestive that CFTR undergoes phase separation with cholesterol domains in the membrane. It undergoes phase, and then coupled to binding interactions from the proteins. And then this also suggests that phosphorylation can regulate the types of clusters that are formed on the membrane. Okay, so I have a bit of time, but in conclusion, the clustering of CFTR requires both cholesterols and protein interactions, and this was supported by our computational predictions and biochemical in vitro data. Calcium or phosphorylation is required for CFTR, and it seems that it's non-exclusive. So you can either have phosphorylation or you can have calcium, and you'll get cluster formation. The clusters are dynamic, and all of this is consistent with phase separation of CFTR and its binding partners on the membrane. And this is possibly one of the more important points I want to make for this session, and this is what we're investigating. So if you have any questions, I'm happy to discuss. So we've derived a couple of models. We have the wild type model on top, and then CFTR, CF-linked variants on the bottom. And so in wild type CFTR, we picture that it's diverse on the membrane, and then upon increases of local calcium, increases of phosphorylation, allowing multivalent partner binding, we get the formation of activity competent clusters on the membrane. And that much of the CFTR activity that we see in cells is actually localized to these clusters. However, when we have a linked variant, we obviously have issues with getting the protein to the cell surface, which is taken care of by correctors. We see that potentiators can increase the conductance of the channels. However, in many cases, as we've previously stated, the expected response to current therapeutics is not what we hope for amongst patients. And so the goals in our lab is there another factor that we need to consider when we think about treating CF? And what we'd like to say is that through the lens of phase separation, we're predicting that we'll be able to develop companion therapies or even other therapies that can drive the rescue of CFTR across various mutants, as well as people that are not responsive to Trikafta to the level that we hope that they will be. And so with that, I'm happy to take any of your questions.
This is great. And in immunology, the LLPSs are already high, so hot. So it's great to see that in ion channel biology as well. So my question is really about the relevance of that to CFTR in terms of its density of expression.
Sure.
So we know, for example, in sweat glands or in ionocytes, this will likely happen. But then a lot of the PDZ domain partners are common to a lot of proteins. So there's definitely competition. What do you think would happen in a cell that doesn't express a lot of CFTR, but CFTR is still needed as opposed to sweat gland or ionocyte?
Yes. So I think there's a couple of things to that point. So we're studying just a small subset of CFTR interactors. We know that the actin cytoskeleton can be involved, et cetera. And so we think that there's probably in various cell types, different mechanisms that can drive CFTR clustering. In terms of the expression levels, CFTR seems to prefer cholesterol-rich domains, although there is evidence that it can exist outside of that. And so what might happen is that it localizes preferentially to cholesterol-rich domains on the surface, which will increase its density in local areas, allowing for function downstream. And so we think that the cells, we also know that CFTR, when there's mutations, it affects cholesterol metabolism. And so it's likely that the proteins themselves are localized to these domains, which kind of predisposes it to be able to form clusters.
So when the cholesterol phase separates, you get cholesterol-rich regions, obviously, but then you also get cholesterol-depleted regions, and so basically the fluid phospholipids also get segregated. Do you know whether your phosphorylated interactions are with the cholesterol crystalline phase or the segregated phospholipid phase? I think you can look at that by having a fluorescently labeled diacyl phospholipid. That will also show the that will show where the lipids are as opposed to the cholesterol. Have you examined that?
Yeah. So we haven't examined that. We actually have the tools to do that, and that's on our list to do pending funding. One of the things that we've noticed in preliminary data that I didn't show is that in cells that express the F508 variant, these are just HEC cells, in which it's been corrected, there's a mislocalization of CFTR to cholesterol-rich domains. So in wild type, most of the detected cholesterol-rich domains that we visualized have wild type CFTR. But in the cells with the variant, there's many cholesterol-rich domains that don't have any CFTR. And so there seems to be some issues with CFTR localization as well. Now, I'm not sure the physical properties of the domains themselves.
So the transmembrane domain also makes extensive cholesterol interaction.
It does.
Very clearly required for stability of the protein, at least in the purified state. You haven't examined that at all yet. Is that right? The interrelationship, interdependency, the R region versus transmembrane region cholesterol interactions.
That's a very good point. We have not. My guess is both contribute. And our models do suggest that both contribute, even though we haven't specifically modeled the cholesterol interactions. Yep.
Okay. Can I ask? So you showed a very elegant sort of system where you've got one membrane, and then on top of that you've got a more complex membrane. So could you sort of apply that to say a bilayer setup in order to do functional measurements of CFTR using, so that your CFTR is is studied under a more sort of native membrane?
In principle, absolutely. The technology isn't quite there yet to measure what's going on in that small interbilayer space. But with that bilayer system, in 2021, we did look at fully transmembrane domain proteins. And it did seem that there was some directionality in terms of membrane insertion into our lipids. So in principle, we can do that. It will require a little bit of further development, but yeah.
I'm sure it's very difficult, but
I mean, I'd like to say that it's easy, but I think the folks that are actually doing the experiments back when I did them would say it's very difficult. Yes.
Would you like to comment?
No, I agree with you. Super.
So Isabelle Sermet, Paris. So thank you very much. It was really beautiful research. Maybe it's a naive question. So because you present data or concept for the plasma membrane, so is your research also applicable for ER membrane, for example?
Yes, it is. And in fact, we developed the system to actually study ER localized sigma-1 receptor. And so the nice thing is that because we can tune the composition of the lipids, we can tune it towards ER. And so we could look to see what's happening during processing of CFTR. Yep.
Yeah, exactly. Co-translational folding. So
exactly. Yes.
Could modulate that also.
Exactly. Yes.
Jonathan, how do you propose that you could modulate clustering as a therapeutic strategy?
Okay, so we have a couple of ideas. Let me just go here. So we're collaborating with David Baker to try to design proteins that can accomplish this for us as tools to actually study clustering. And then another thing that we've been thinking about is how to modulate.
Could you go into that a little bit? Because I'm not sure.
Okay, so being in Seattle, I mean, there's something there, right? But David Baker, just down the road here, University of Washington, computationally designs proteins, binders that are not found in nature, but that can interact with existing proteins. And so, working with him and his postdoc Keja, we've designed. She's designed thousands of binders that can interact with the R region in various locations. And so we're now testing those predicted binders to see if we can, number one, bind to CFTR in areas that are not currently bound by binding partners. So this would be a new interaction space. And then use that multivalent binders that can then be expressed in cells and can artificially cluster CFTR without interfering with the native biology of the receptor. And then we're thinking about how to manipulate cholesterol localization using extracellular entities. So can we take just these small cholesterol-rich domains and then reorganize and then force them into larger domains, like we see with CFTR?
Okay, well, thank you very much.
All right, thank you.
It's my pleasure to introduce my co-chair, who has kindly stepped in to do some pitch hitting because unfortunately Dr. Vergani can't be here today. Dr. David Shepard will be speaking about single-channel studies of CFTR delta 508 rescue modulators and how they interact with function of delta 508, elexacaftor, tezacaftor, and ivacaftor. Oh, sorry.
So, thank you very much, Christy. So we're all very sorry that Paola can't be here to deliver the presentation, and we wish her and her family all the very best. So, just to qualify what Cedric was saying in his presentation, I didn't do any of the experiments. My eyesight is not good enough to do patch clamp, and so I really need to sort of say a huge thank you to my current and past colleagues. So the experiments that are presented in this presentation and those that you saw in Cedric's presentation were undertaken predominantly by James and Myri. Then in this presentation, some data from them. Okay, so we know that CFTR is synthesized in the endoplasmic reticulum, matured in the Golgi, and then delivered to the cell surface where it forms a anion channel with complex regulation. And then F508del has multiple mechanisms of CFTR dysfunction. So first and foremost, the misfolding prevents the protein being delivered to the cell surface. But if you can coax the protein to the cell surface, such as by low temperature incubation, then you would see two further defects. So reduced stability at the cell surface and defective channel gating. So to overcome those defects, we need correctors to deliver the protein to the cell surface, correctors to stabilize the protein at the cell surface, and then potentiators to enhance its activity. So what I'd like to discuss with you today is the single channel studies that we undertook to investigate how elexacaftor, tezacaftor, ivacaftor, how they affect the single channel behavior of F508del using excised inside-out membrane patches from cells expressing recombinant CFTR. So The presentation will have two parts. So first we look at the rescue of F508 by the triple combination therapy. And then second, some mechanistic studies that James and Myri undertook with alexicaftor. Okay, so to sort of orientate you into how the majority of the experiments were undertaken in this study is we have the baby hamster kidney cells that Margarita generously provided us with that are expressing F508del. These cells are treated with alexicaftor, tezacaftor, ivacaftor. So here's the concentrations: 2 micromolar of alexicaftor, 3 of tezacaftor, and 1 micromolar of ivacaftor. And this chronic incubation for 24 hours at 37 degrees C is the drugs are in the presence of culture media, which contains fetal bovine serum. So then after that incubation period, the cells are washed to remove the culture media and the FBS. And so then the cells are in a physiological salt solution that's high in chloride, and that's used for our patch clamp experiment. So we have a high concentration of chloride on the intracellular side of the membrane patch, low concentration of chloride on the extracellular side in the patch pipette. And so what we'll see, the downward deflections of the traces that I show you correspond to chloride moving from the intracellular side of the membrane to the extracellular side of the membrane in the patch pipette. And then all the recordings are being made in the presence of ATP, 1 millimolar, and protein kinase A. Okay, so here shows you the representative examples of single channel recordings of wild type CFTR at the top, F508del then rescued by low temperature incubation, and then the pairs of CFTR modulators, so alexicaftor, tezacaftor, tezacaftor, ivacaftor, alexicaftor, ivacaftor, and finally the triple combination alexicaftor, tezacaftor, ivacaftor. So to orientate you in other single channel recordings, so we have single channel current on the y-axis and time on the x-axis. The dotted line shows you where channels are closed, and these downward deflections of the trace correspond to chloride moving from the intracellular side of the membrane through, in this case, single channels to the extracellular side of the membrane in the patch pipette. On the right-hand side, you're seeing what are called current, you're seeing current amplitude histograms. And so for wild type CFTR, this narrow tall peak is corresponding to the closed channel level, and this smaller, fatter peak, it corresponds to the open channel. The distance between the two peaks corresponds to the amount of current that flows through an open channel. And so these recordings then are made initially at 37 degrees C until channel activity. These recordings are representative of what we observed before channels deactivated. And that will occur after different lengths of time, depending upon the different combinations of modulators that we've used. And so just looking at the patterns of opening and closing, you see that they're very different from wild type CFTR, and even the triple combination therapy here, where we've chronically treated the cells with alexicaftor, tezacaftor, ivacaftor, but now the recordings are made in the presence of ATP and pKa alone, you're not restoring the wild type level of activity. So we're quantifying channel activity by measuring the size of the downward deflection and the pattern of opening and closing, which is open probability. So when you do that, so the current amplitudes are mostly similar to that of wild type CFTR for F508del, except for the F508del that has been rescued by the combination of tezacaftor and ivacaftor, where you're seeing a small reduction. When you look at the open probability values, so open probability or PO for short, this is a measure of the average fraction of time the channels are open. So you see that in the case of the tezacaftor ivacaftor treatment, and these are the recordings immediately after you've rescued, after you've started the recordings, after you've added pKa and ATP, you're seeing levels of open probability equivalent to wild type CFTR. But for the other combinations, including elexacaftor, tezacaftor, ivacaftor, chronically incubated cells, you're not recovering open probability to wild type levels. So our first conclusion then is that combinations of modulators have distinct effects on the single channel behavior of F508del. The current amplitude of CFTR, F508del CFTR rescued by ETI is similar to that of wild type CFTR, but the open probability of ETI rescued F508del is reduced compared to that of wild type CFTR. Okay, so to look at the changes in open probability in a little bit more detail, James and Myrie, or in this case, Myrie, measured the duration of channel opening. So that's mean burst duration. So if I just go back and let's just so we're going to look at two characteristics. So mean burst duration is going to be the average duration of a single channel opening. And then interburst interval, or IBI, is the duration of the long closure between one opening, one burst of opening, and the next burst of opening. So if we come back to the summary data here. So here's our open probability data. And if you look at the duration of the openings or the duration of the closings, you're seeing sort of variable effects corresponding to the variable effects on open probability. So you see that our sort of the duration of openings of low temperature rescued are less than those of wild type. In the case of elexacaftor, tezacaftor, there's about equivalent to those of wild type, whereas the other duration of openings appear to be increased, particularly the tezacaftor, ivacaftor treated channels. The duration of long closures separating one opening from the next opening, you see that in all cases, the duration of the long closures are much longer than those of wild type CFTR, particularly for this elexacaftor, tezacaftor treatment. Okay, so combinations of modulators have distinct effects on the pattern of opening and closing. They modify the frequency and duration of channel openings, and ETI does not restore either the open probability or the gating pattern when it's used chronically alone. So to sort of further study the effects of this chronic treatment with elexacaftor, tezacaftor, ivacaftor, Myrie performed these prolonged experiments. So here you're looking at an F508del CFTR excised inside membrane patch from a cell that's been treated with a combination therapy for 60 minutes in duration. Now, I'm not showing you a recording with low temperature rescued F508del, but we would never be able to sustain a channel activity for this length of time if we rescued by low temperature. So this was a really very marked observation as to how the F508del channels were behaving when they were treated, when cells were chronically treated with elexacaftor, tezacaftor, ivacaftor. So to sort of orientate you here, so the arrowheads correspond to different levels of channel activity. So we have one channel here, second channel here, and so we've got at least 3 channels active here. And then you can sort of see the pattern of opening and closing on a sort of a higher resolution at the beginning of the recording, in the middle of the recording, and at the end of the recording. So even at the end of the recording, we've got substantial channel activity for F508del. So the rescue by the triple combination therapy means that we have stable channel activity, and the channels deactivate much, much more slowly in excised cell-free membrane patches compared to those of channels that have been rescued by low temperature incubation. So if we look at the effects of current amplitude and open probability, here's the current amplitude data for the different combinations. So here is the current amplitude for wild type CFTR. What we're doing in this slide, in the next slide, and in some of the subsequent data I'll show you, is that we're measuring current amplitude or open probability over the first 5 minutes of the recording, over the 5 minutes at 25 to 30 minutes, and then at the final, the 55 to 60 minutes. And so you see that for cells that have been chronically treated with the triple combination therapy, you have stable current amplitude, whereas if we have low temperature or these pairs of CFTR modulators, we don't have stability of current amplitude. And then if we look at the open probability data, so I drew your attention to this sort of dramatic enhancement of channel activity when you have tezacaftor, ivacaftor alone, but that dramatic activity is very short-lived, and so over a prolonged period, we very rapidly lose channel activity when you have cells that have been rescued by just tezacaftor and ivacaftor. In the case of elexacaftor, tezacaftor, or elexacaftor, ivacaftor, so you see that again you have loss of channel activity over time. Only when we have cells that have been treated with the triple combination therapy do we see sustained channel activity, albeit that level of activity is not wild type level of activity. These recordings are made in the presence of ATP and pKa alone. So to summarize then, single channel current amplitude, so the amount of current flowing through an open channel rescued by the triple combination therapy, is stable in cell-free membrane patches and similar to that of wild type CFTR. The open probability again is stable, but it's reduced compared to that of wild type CFTR. So let's move to the sort of second part, the mechanistic studies. And so here, the first thing that we did, and so what Myri has done here is she's kept the concentration of tezacaftor and ivacaftor constant, and she's varied the elexacaftor concentration that the cells have been incubated with. And so here you're seeing the representative channel recordings. And so if we look at the recording at the top here, where the cells have been incubated with 0.1 micromolar elexacaftor in the presence of 3 micromolar of tezacaftor and 1 micromolar of ivacaftor, you look at the size of the downward deflections are smaller compared to the size of the downward deflections when we use either 2 micromolar elexacaftor or 10 micromolar elexacaftor. And that's sort of more clearly seen in the distance between the closed and the open current levels in the current amplitude histograms when we compare here for 0.1 micromolar of elexacaftor with either here or here, where we've got 2 micromolar or 10 micromolar of elexacaftor. Okay, so here's the quantification, and you can see that the current amplitude is increasing with increasing elexacaftor concentrations. If we look at the open probability data with elexacaftor 10 micromolar, you appear to have a higher open probability. So raising the elexacaftor concentration increases single channel current amplitude, and then elexacaftor 10 micromolar appears to increase the open probability by prolonging the duration of openings. So then what about the stability? And so here the plots are summarizing the current effect on current amplitude and open probability. And so if we look at the data for current amplitude, and we've got wild type on the left here, you see that for 2 micromolar, we're stable at either short time intervals, intermediate time intervals, or prolonged time intervals. Whereas for lower concentrations and for the higher concentration of elexacaftor 10 micromolar, you see that with prolonged time intervals, you have instability of channels, and you have a reduction in current amplitude. Correspondingly, the open probability, while it was at 10 micromolar, while it was increased at short time intervals, and at prolonged time intervals, you had a loss of channel activity. So in the case of these baby hamster kidney cells that have been chronically treated with elexacaftor, tezacaftor, ivacaftor, we would argue that elexacaftor 2 micromolar is the key concentration to rescue in this particular cell preparation, where you've got stability of current amplitude and stability of open probability. So only using elexacaftor 2 micromolar was channels stable, and potentiation by the 10 micromolar of elexacaftor was transient. Okay, so we've got stability of the open channel structure of F508 del would be the interpretation of the results so far. So what James was very interested to test was the acute effect of elexacaftor. And to test the acute effects of elexacaftor, we turned to recordings of wild-type CFTR. And so what you're looking at here is a control recording of wild-type CFTR in an excised inside-out membrane patch in the absence of elexacaftor, but presence of ATP and pKa. And then the recording, the channel traces after elexacaftor 2 micromolar has been acutely added to the solution bathing the intracellular membrane patch. And so if you compare the channel traces, you see that in the presence of 2 micromolar of elexacaftor, the channels appear to be more active than in the absence of elexacaftor. And if you look, if you compare the distance between the closed current amplitude and the opened current amplitude in the absence and presence of elexacaftor, then there is a small increase in current amplitude in the presence of elexacaftor. So to quantify these data, so James measured single channel current amplitude. His open probability data are here. And in the middle, you've got the duration of the long closures and the duration of those channel openings. And so if you look at the pattern of opening and closing to begin with, you're seeing an enhancement of the open probability of wild-type CFTR. And those results are consistent with previous data showing that elexacaftor but not only corrects but also has potentiator activity. And that effect, that increasing open probability, is mediated by an increase in the frequency of channel opening, so a reduction in the interburst interval, and an increase in the duration of channel openings by this increase in mean burst duration. But what we were really sort of struck by was that there was a consistent increase in current amplitude of wild-type CFTR in the presence of elexacaftor. So the effect is small, and I'll be the first to say that, but it's a consistent and it was statistically significant increase in current amplitude when you acutely treat wild-type CFTR with elexacaftor. So based on these data, James was keen to see, okay, I've got ahead of myself. This was going to happen at least once. So elexacaftor then, to summarize, elexacaftor strongly potentiates wild-type CFTR by enhancing the frequency and duration of channel openings, and elexacaftor causes a small but statistically significant increase in current flow through open channels. Okay, so then James was then very sort of keen to sort of test, well, what happens to F508del? And so to sort of orientate you here, what James has done, he's taken the F508del. So we're looking at the sort of second trace here. So what James has done is he's taken his F508del expressing BHK cells that have been chronically treated with elexacaftor, tezacaftor, ivacaftor, and then he's excised an inside-out membrane patch. And that membrane patch is now being exposed to ATP, pKa to activate channel activity. But very importantly, he's got 2 micromolar of elexacaftor, and in this case, 100 nanomolar of ivacaftor in the intracellular solution. So you've got both chronic treatment of the cells with elexacaftor, tezacaftor, ivacaftor, and then acute treatment with elexacaftor and ivacaftor. And so your current amplitudes are about the level of wild type. But here now, you're instead of an open probability value of the chronically treated cells with the chronically treated F508del cells with elexacaftor, tezacaftor, ivacaftor having a reduced open probability compared to wild type. Now, if you have those chronically treated cells plus acute treatment with elexacaftor and ivacaftor, you have an open probability that is, if anything, is larger than the open probability of wild type CFTR. But you're not restoring, so this pattern of opening and closing is not the same as wild type CFTR. So your duration of the long closures separating one channel opening from the next is larger than that of wild type CFTR, while the duration of the channel openings of rescued F508 under these conditions is much, much larger than those of wild type CFTR. Okay, so then to summarize then, acute co-treatment with of with elexacaftor and tezacaftor restores wild-type like channel activity. It's not exactly the same, but it's approaching that of wild-type when F508del has been chronically treated with elexacaftor, tezacaftor, ivacaftor. So to try and sort of schematicize this then, so we need our correctors, elexacaftor and tezacaftor, to overcome the misfolding of F508del and deliver the protein to the cell surface. We need elexacaftor and tezacaftor to stabilize the protein at the cell surface. And then we would have ivacaftor and elexacaftor to enhance channel activity at the cell surface. But what we're really excited about is the fact that elexacaftor is also sort of stabilizing the channel pore and allowing, in the case of wild-type CFTR, enhancing the amount of chloride that is flowing through an open channel. So we would sort of argue, we would interpret these results to suggest that elexacaftor is a triple-acting CFTR modulator, which augments not only the expression of CFTR, but its gating behavior and the current flow through an open channel. So the triple combination therapy is in this sort of model system is restoring stability and I've got to modify this, but I didn't work. Okay, it's restoring the stability and it is restoring the function. It's restoring wild type like function. So I apologize for this incorrect title. We only could change so much this morning. Okay, thank you very much for your attention and bearing with me. Right? Okay. Let's see.
Thank you. Well, people are lining up behind the microphone.
Okay. Yeah.
I had the impression that when you looked at elexacaftor acutely after correction on delta 508, that the conduction was decreasing rather than.
So what we're doing then is what we've got is two sets of data. So one set of data that Myri undertook, and then a second set of data that James undertook. And yes, you're right that it's one is smaller than the other, but if you take James's data, then they are sort of equivalent to wild type. I think that would be my key point, is that that the current amplitude data are equivalent to wild type, but I fully take that point that there is a difference between the two sets of experiments. Yeah. Thank you, Christine.
So, David, at this point, there are a number of compounds that are known that bind to that site. Have you compared different compounds binding to that site to see whether they're all those?
So, sorry, Jay. Sorry, John. That is the extent of what we've done so far at the single channel level, right? I think it would be wonderful to sort of compare and contrast, but we've not done so. Sorry.
Okay. Martina Gensch, UNC Chapel Hill. Great presentation, David. I'm happy to say that Andrei Alexandrov, who has retired in the meantime, had data in lipid bilayers that are very complementary to yours. So elexacaftor was preventing inactivation of rescue delta 508. Now, what I'm wondering is, also as a cell biologist, these lipid bilayers and also your system is like cell-free. And we always talk about, stabilization, but there are so many kinds of stabilizing CFTR, the function, the structure, then preventing it from degradation. Do you have any thoughts on how that goes together? Like, do you think the destabilized channels are the ones that are then internalized, or like basically relating your data to cell surface stability?
So this is a very important point, Martina. So what we see when we rescue F508del, not with the triple combination therapy, with, say, if we look at our own data, which was predominantly with low temperature incubation, what you would see is over time, you see a reduction in open probability, but you also see the current amplitude decreases over time. And so how we interpreted those data were that the channels were basically falling apart before your very eyes. Now, if we used, and again, thank you, Margarita, if we used the baby hamster kidney cells that were expressing F508del with revertant mutations, there we could see stability, that we didn't have the loss of channel activity. So you had stable current amplitude and you had stable open probability measurements. So I think that what we see in an excised cell-free membrane patch is that the channel is falling apart before our very eyes. Now, so sort of to tell you what I had an argument with Linda Ostergaard about these data, and Linda sort of interpreted these data to sort of say that, well, David, I think you've got sort of internalization even in an excised inside-out membrane patch, but I just don't know if that would be the case. What I can say is that, and we didn't show the data, is that Myri not only did all the sort of single channel studies, but she's also used Ussing chamber studies. Those were with the FRTs that were expressing F508del hydrolycously and treated with the same combinations that I showed you at the single channel level. And our Ussing chamber data mirrors our single channel data. So with the triple combination, you have stability. Okay, we only did it over 8 hours with the cells that were treated with cyclohexamide to just measure the population cell surface. But we did see similar results with the FRTs compared to the single channel data.
Awesome.
Thank you.
Did it quite a tour de force what you are presenting here. Like, I'm glad I didn't have to do all the patching for that, but I was struck, particularly when you tested the combinations in your first part, that really you saw the largest open probability from IVA and TESA rather than sort of the triple combination, when obviously in the clinics there's no doubt that the triple combination is the most efficacious. And one thing that struck me is that you're using basically for your chronic incubations the 1 micromolar of either Kafta or Martina, and Gary Lukács has also shown that a destabilized CFTR or something. And
so, did you want to say more? Or?
Well, I mean, I was just wondering, like in your later part, obviously you had some reasons, the channels lasting longer, where I can maybe get back to that, the triple maybe working better, but I'd like to hear your thoughts.
So I apologize if I didn't explain it clearly enough. So those initial single channel recordings where you saw the different combinations, what Myri did is she took the first 30 minutes of those 60-minute recordings and just quantified open probability or current amplitude over that time period. Yeah. And then to measure stability, instead of trying to do it over the entire recording length, we just took the first 5 minutes, the middle 5 minutes, and then the end 5 minutes. And so now I'm going to get myself confused. So with the orange bars, that you saw this very high open probability to begin with, but it just didn't last. Yeah.
So that's the explanation basically why.
Yeah. And just to sort of repeat then, so the baby hamster kidney cells are treated with the different combinations in culture media in the presence of FBS. So, we haven't done it plus or minus FBS, but I think that is probably the explanation.
Yeah, but FBS is 10% or something, right? So it's far from human serum.
Well, okay, but it's hand waving. These are the experimental conditions we've used.
Yeah, no, I was just particularly the Iva Kafta concentration. Like we typically, when we do chronic, we only go like 50 micromolar or 100 micromolar because you're already saturated.
Yeah.
But then once you don't have it present, once you excise your patch, it's hard to control what actual concentration you're really.
Exactly, that you wash, but you're not going to get the membrane patches are going to be saturated with the compounds, and you're not going to be able to get the compounds out of the membrane patches. Yeah, it's a very good. Yeah.
Okay.
Last question. So very nice work, a lot of work. I try to learn patch clamp, but just too much. So what's your consideration of using the hamster kidney cells? HBV.
So sorry, why did we use?
Yeah, why you choose this one?
They're just great cells to sort of. Thank you, Martin. So Single channel recording. So when we have stable cell lines, we prefer to use the stable cell lines, providing you can make the measurements. So some stable cell lines will have too high a level of expression to do single channel recording. But the baby hamster kidneys with F508del seem to work very nice. So that's how we would do our F508del studies. If we're doing variants that we don't have stable cell lines for, we'll do transient transfections. And for those, we prefer to use Chinese hamster ovary cells, because the Chinese hamster ovary cells have a very sort of silent, they have a relatively silent background. So there's less contaminating channels, so that's always a sort of issue is, are there contaminating channels? And,
I see.
In the days of old, when I did the experiments, there would be a lot of cursing and swearing when the contaminating channels appeared, right?
Okay, I'm thinking, CLPE cells, kind of things.
Sorry.
The CLPE more.
They're just too difficult to patch, right? Just don't even think about it, right?
One quick one, TC.
Okay. Oh, yeah, I want to follow up Martin's question. In the first part of your talk, your recording, if I understand correctly, your recording was made in the absence of any drugs, right?
So you've got ATP, you've just got ATP,
right,
PKA, but no drugs.
Right. So the pre-treatment is like before you make a recording.
It's for 24 hours at 37 degrees C, wash the cell, and then make the recording.
Then the lower open probability you observe with ETI rescue delta F508, wouldn't that just because of washout of ivacaftor? Right? Because later on in the second part of your talk, you say by acutely adding back ivacaftor and elexacaftor, the PO increase.
Yeah. That was so what we haven't done, and so Martin, I might need your help, right? So currently we have patch clamp rigs in pieces and we don't have any PKA, but when we have patch clamp setups in working order and we have PKA, the first experiment James wants to do is to do the experiments of adding either ivacaftor or elexacaftor acutely and then seeing how much channel activity you see. But I agree, it's a very good point.
Right.
Well, I'd like to thank all of the speakers for a very stimulating session and wonderful discussion by the group here. Thank you.